Welcome to the deep dive. Today, we're really getting into something fundamental. It's happening in you right now, constantly. Cell division.
It really is an incredible process, almost like a hidden world inside us.
Exactly. And usually your cells follow this really carefully choreographed routine. We call it the cell cycle. It's like a four-act play.
Right.
First up is G1. That's when
Yeah.
the cell grows, gets bigger, and sort of preps its DNA for copying.
Getting everything ready.
Then you have the S phase, S for synthesis. That's the main event for DNA copying. Replication happens
The
here.
crucial duplication step.
Next is G2. Here the cell double checks everything, looks for DNA errors, gets ready to actually divide.
Like a final quality control check.
And finally, M phase, mitosis. One cell splits gracefully into two identical daughter cells. Perfect copies.
Hopefully perfect, yes.
And here's what's really amazing, I think. Between these acts, there are these like built-in checkpoints.
Ah, yes. The gatekeepers.
Like super strict bouncers, making sure everything is absolutely spot on before the next step. I mean, this system catches and fixes millions of potential problems daily.
It's incredibly robust.
And if it finds damage, it can't fix. It can pause the cycle for repairs or, well, it triggers apoptosis,
Programmed cell death. The cell essentially takes one for the team.
removes the problem before it spreads.
The precision is just, well, staggering when you think about it. That normal cycle, those safeguards, they're so sophisticated.
Yeah.
But, and this is really where our deep dive starts today, cancer cells, they don't just bend the rules. They seem to completely shatter them.
Shatter them how?
They hijack the system, break those rules. They essentially blow past those checkpoints you mentioned.
Ignore them.
Pretty much. Which leads to, that uncontrolled growth we see as tumors. It can start with just a few key failures, really, a few broken parts in that precise machine.
Wow. So it sounds like this system should be almost foolproof. How do cancer cells manage this checkpoint failure? Do they just brute force it, or is it sneakier?
Oh, it's definitely insidious, very sneaky.
Yeah.
You have these really important genes, tumor suppressor genes, maybe you've heard of P53 or ARB.
Yeah, I've heard of P53, the guardian of the genome, they call it.
Exactly. Normally, they act like brakes. They stop the cell cycle if something's wrong, maybe trigger that apoptosis we talked about.
Okay.
But in many cancer cells, these genes get switched off, inactivated. So the brakes are gone. Damaged cells, they don't get repaired, they don't die. They just keep going, keep dividing.
So the warning lights are ignored, and the car just keeps speeding up.
That's a good analogy. And maybe even more startling, cancer cells figure out how to replicate almost indefinitely. They switch back on an enzyme called telomerase.
Telomerase, that rebuilds the ends of chromosomes, right? Telomeres.
Precisely. Normal cells run out of telomere length after a certain number of divisions, which forces them to stop. It's like a built-in counter. Cancer cells reactivate telomerase, rebuild those ends, and effectively, well, they become immortal. They bypass that counter.
Immortal cells, that's quite a thought. Almost like cellular vampires dodging death.
It is a chilling concept when you frame it like that. They escape the normal limits of cellular life.
Okay, so if they've disabled the brakes and become immortal, how does that warp the whole dance, that meticulous four-act play you described earlier? What does the cycle actually look like in a cancer cell?
It's completely distorted. Imagine cells that should pause in G1 because they're damaged or not ready. They just barge right through. No pause for repair.
So damage just accumulates right from the start.
Exactly. Then the S phase, which should be about making perfect DNA copies, it becomes, well, almost a mutation factory because the initial DNA might be damaged and the copying process itself can be error-prone.
More mistakes piling up.
Then in G2, where the cell is supposed to double check for errors, that damaged, mutated DNA often just gets waved through. Checkpoint failure again.
So no final inspection.
Nope. And the result. M phase becomes chaotic. Rapid, abnormal cell division. Cells dividing with the wrong number of chromosomes, full of mutations, leading to that relentless tumor growth. It's like the Wild West compared to the usual order.
Wow. So understanding this broken cycle, what does that actually mean for us, for you listening? How does it connect to treating cancer?
It connects directly. I mean, many common treatments, like chemotherapy and radiation, they work by specifically targeting cells that are dividing rapidly.
Ah, because cancer cells are defined by that rapid, uncontrolled division.
Precisely. They're often caught in the S phase, copying DNA, or the M phase, actually dividing. The treatments hit them hardest then.
Which also explains the side effects, right? Because other cells in our body divide quickly, too.
Exactly. Think about your hair follicles or the lining of your gut. They divide normally quite fast. That's why those therapies can unfortunately damage those healthy, fast-dividing cells too, leading to common side effects like hair loss or nausea.
Makes sense. A bit of a blunt instrument sometimes.
It can be. And that really highlights the huge challenge, doesn't it? The ongoing research is all about finding ways to be more specific. How do we target only cancer's broken runaway cycle, like a laser beam,
Yeah.
while leaving all the healthy, normal cell division processes untouched, protecting the vital functions that keep us alive and well?
A massive puzzle.
It truly is. It makes you really think about, well, the incredible complexity and intelligence built into our bodies and the sheer ingenuity required to try and outsmart something that's gone so fundamentally wrong at the cellular level. How do you fix the rules when the rule breaker is part of you?
A profound challenge, and definitely something to keep thinking about.